You know, your immune system is pretty incredible. It's this microscopic army working around the clock to protect you. But what happens when that army gets its signals crossed? When it overreacts or attacks the wrong target? Well, that's when good immunity goes bad, and we call it hypersensitivity.
So think about it like this. Whenever your body faces some kind of threat, the immune response can really go one of three ways. It can be just right, totally adequate, and you heal up. It can be inadequate, which leaves you vulnerable. Or it can be excessive, a massive overreaction. And that's exactly what we're diving into today.
So when an injury or an invader shows up, you see these different paths it can take. The ideal path, of course, is that adequate response leading to healing. But look at this other branch, the excessive or misdirected response. This is where hypersensitivity lives. It's basically the body using a sledgehammer to kill a fly, and the collateral damage is the real problem.
So here's our game plan. We're going to visually walk through the four main types of hypersensitivity. We'll map out the step-by-step pathophysiology so you can really see how each one works inside the body.
All right, let's start with a quick, high-level look at these four reactions. Okay, this table right here, this is your ultimate cheat sheet. It lays out all four types, what's driving them, and a classic example for each. So notice the main culprits. Type I is all about an antibody called IgE. Type 2 involves antibodies that target our own tissues. Type 3 is about these floating clumps called immune complexes. And Type 4, that one's run by T cells. Keep this framework in your head as we break each one down.
Okay, let's dive into Type 1. This is the one most of us are familiar with. We're talking everything from seasonal allergies to food allergies and bee stings. This is the classic allergic reaction. This image perfectly captures the two-act play of a Type 1 reaction. The top half is what happens the first time you're exposed to an allergen. The bottom half is the drama that unfolds every single time after that. Let's start with Act 1, the first exposure.
So get this, the very first time you encounter something like pollen, you feel absolutely nothing. But inside, your body is quietly preparing for war. Your T helper cells, which are like the generals of the immune system, tell the B cells, your antibody factories, to start cranking out a special antibody called IgE. This IgE then goes and sticks all over your mast cells, which are basically little grenades packed with chemicals. The grenades are now armed. You're sensitized, and you don't even know it.
So what happens the next time the pollen gets in? Well, now it finds those armed and ready mast cells. The pollen clicks into those IgE antibodies, and that's the trigger. The mast cell degranulates. It just dumps its entire chemical payload, especially a ton of histamine, into your system.
And this is what that chemical flood looks like on the outside. All that histamine makes your blood vessels leaky. Fluid seeps out from the vessels into your skin, and you get these itchy, raised welts we call hives, or urticaria. You are literally seeing the fallout from those mast cells exploding.
All right, let's switch gears to Type 2. Now we're moving from an overreaction to a foreign substance to a case of friendly fire, where the immune system starts targeting the body's own cells in very specific tissues.
The key thing to visualize here is an antibody physically latching onto an antigen that's part of one of our own cells, like a red blood cell. It's essentially slapping a giant "Destroy Me" sticker on a perfectly healthy, normal cell. So, once that cell has been tagged for destruction, how does the body actually take it out? Well, it turns out there are a few different strategies, and we're about to look at the three big ones.
So, the body has a few options. First, it can unleash the complement system. Think of this as a demolition crew that just punches holes in the cell until it bursts. Second, it can call in the cleanup crew, cells like macrophages that literally come and eat the tagged cell. Or third, and this is really interesting, it can just mess with the cell's function by using antibodies to either block its signals or jam them into the on position.
Graves' disease is a perfect example of that third mechanism in action. Here, an antibody doesn't destroy the thyroid cell. Instead, it impersonates a hormone called TSH. It binds to the receptor and just holds down the go button, forcing the thyroid to work overtime and pump out way too much thyroid hormone.
Okay, on to Type 3. The problem here isn't antibodies attacking a cell that's fixed in one place. Instead, you have antibodies binding to soluble antigens, stuff just floating around in your bloodstream. They form these clumps called immune complexes that can cause major problems wherever they end up lodging. Now, this is a really important distinction, especially for exams. Think of it this way. Type 2 is like a sniper,
taking out a specific stationary target. Type 3 is more like pouring sludge into the body's plumbing. These clumps flow everywhere and just clog up the works wherever they get stuck, especially in delicate filters like your kidneys and joints.
And this is what that clogging actually looks like. Those bright yellow clusters, those are immune complexes that are literally stuck inside the glomerulus, which is the kidney's tiny filtering unit. Once they're stuck, they trigger a huge inflammatory response that damages the filter itself. This is exactly what we see in conditions like lupus nephritis.
Finally, we have type 4. This one is the odd one out. It's the only one that doesn't involve antibodies at all. Instead, it's run entirely by T cells and, true to its name, it's always fashionably late to the party.
So the defining feature here is the delay. Why the wait? Well, it's because this reaction isn't driven by pre-made antibodies that are ready to go. It takes time for the right T cells to get activated, actually travel to the site of the problem, and then recruit their backup, like macrophages, to launch the attack. It's a much slower process.
The absolute classic example to help you visualize this is poison ivy. The first time you touch it, nothing happens. But your T cells are taking notes, they're learning to recognize that oil. The next time you brush up against it, those memory T cells spot the antigen in your skin, they swarm the area, and they release chemicals that basically start a war. And it's the damage from that battle that causes the itchy, blistering rash a day or two later.
So let's do a quick final roll call of our main culprits. For type 1, it's all about IgE and those exploding mast cells. For type 2, it's antibodies attacking our own tissue. For type 3, it's those circulating, sledge-like immune complexes. And for type 4, it's the delayed response from T cells and their backup. 4 very different ways the immune system can go wrong.
Understanding these pathways is, of course, the first step, but it leaves us with a really critical question when it comes to treatment. Where is the best place to intervene? Do we try to stop the initial trigger? Do we block the specific immune response itself? Or do we just focus on cleaning up the inflammatory mess afterward? Finding the best answer is the key to managing all of these conditions.